Morphogenesis, creation of 3D form. It is shaping of the multicellular body and its organs. Pattern formation, morphogenesis. Pattern formation is the mechanism by which initially equivalent cells located at a particular position in a developing tissue of the embryo assume complex form and functions. Arrangement of equivalent cells of particular lineage in developing embryo which will decide its shape and function based on its position. Cell movements occur in pattern formation and morphogenesis. The first step in pattern formation is the establishing of body axis. Three types of axis: anterior posterior, head to tail, dorsal ventral, back to front, and right and left axis. हेड की तरफ एंटीरियर, टेल की तरफ पोस्टीरियर, वेंट्रल फ्रंट, डोर्सल बैक. Next stage is gastrulation, formation of three different germs layer, ectoderm, mesoderm and endoderm. Further, the cells of three germ layers acquire different identities based on their position. The position of cells in the germinal show their prospective fate. Two general modes of pattern formation. First, use of morphogen gradient. Second, by sequential embryonic induction. Morphogenic gradient. Morphogen is defined as a signaling molecule that acts directly on cells to produce specific cellular responses dependent on its concentration. It can be cytoplasmic factor, example bicoid, caudal, nanos, gradient in multinucleate cell. Secreted molecule. Second is secreted molecule like retinoic acid into cell surrounding. Lewis Wolpert, concept of morphogen using French flag model. Cells can form blue, white, red. Cells can't form American flag whatever position you change. Different tissue may use the same gradient system but respond to the gradient in a different way. Reciprocal transplantation. Cells from blue region of French flag transplanted to striped region of American flag, then cells will form structure of French flag depending on morphogen gradient at that place. Since new region where cells are placed receive low morphogen gradient, then the cells will form red structure of French flag even when placed in American flag. Piece of tissue transplanted from one region of an embryo to another retains its identity but differentiate according to its new positional instructions. Morphogen gradient. Drosophila, antenna and leg respond against same morphogen but form organ specific structure. जहाँ पर ज़्यादा मोर्फोजन है उधर बनेगा लेग के केस में कोक्सा, फिर कम उससे मीडियम मोर्फोजन वाले एरिया में टीबिया और कम वाले में क्लोज. कोक्सा, टीबिया, क्लोज. और एंटीना सबसे ज़्यादा मोर्फोजन होगा तो पेडिसल, मीडियम होगा तो एंटीनल सेगमेंट्स एंड फ्यूनिक्यूल्स और कम मोर्फोजन होगा तो एरिस्टा, फ्लाई एंटीना. पेडिसल, antennal segments and funiculus and arista or fly antenna. Examples of morphogen. First, vertebrate body axis or limb determination, retinoic acid, FGF8 and WNT. Second, formation of regions of mesoderm of limb by bone morphogenetic protein BMP. Third, formation of vertebrate limb axis and digits by sonic hedgehog. Embryonic or sequential induction. Influence of one cell or group of cells on adjacent cell, resulting in the change of cell behavior like change in shape, mitotic rate or cell fate. Inducer cell send signal and competent cell receive signal. Inducing cell can be paracrine or juxtacrine. Competence is the ability to respond against inductive signal. Receptor must be present for that inducer. Example of embryonic induction: vulva formation in C. elegans. Signal 1, when received signal 1 then form primary vulval cells. When signal 1 and 2 is received, then secondary vulval cells. When no signal received, then default fate hypodermis. Cell death: accidental cell death, necrosis like disease, injury, virus or bacteria induced. Second is programmed cell death, PCD. Two types of apoptosis and autophagy. Apoptosis, developmental, morphogenesis, cell number, balance. ऑटोफाजी इन स्ट्रेस कंडीशन. एपोप्टोसिस में क्या होता है? सेल श्रिंक, डीएनए फ्रेगमेंटेशन, न्यूक्लियर फ्रेगमेंटेशन, एपोप्टोटिक बॉडी फॉर्मेशन, क्लियरेंस बाय मैक्रोफाज, प्रोग्राम्ड मैनर. Only unwanted cells are removed from specific location. No cytosolic content is released outside. Reproduction: the biological process by which new individual organisms, offspring, are produced from their parent or parents. Asexual reproduction, single parent. Sexual reproduction, two parents. Asexual reproduction, single parent is involved. Progeny are clone, genetically identical to parents. Seen in both unicellular and multicellular organisms, prokaryotes and eukaryotes. Progeny are produced by mitosis and cytokinesis. Chromosomal separation and cell division in prokaryotes. No fertilization or gamete formation take place. No meiosis. Example: binary fission in bacteria. Fission yeast, Schizosaccharomyces pombe, and budding in budding yeast, Saccharomyces cerevisiae. Budding में क्या होता है? इक्वल न्यूक्लियर डिवीजन बट अनइक्वल साइटोप्लाज्मिक डिवीजन. Fragmentation in hydra and bacteria, sporulation in bacteria and fungi during stress, Bacillus subtilis. Plants' asexual reproduction is known as vegetative propagation. Benefits of asexual reproduction: no mate is required, rapid proliferation, progenies are clones or identical. Disadvantages: no variations, suitable only constant environment, all survive or none will survive. Extinction prone. Sexual reproduction: two parents are involved, gametes are produced by meiosis. Very high variations among progeny. Variation reasons are independent assortment, depends on arrangement of homologous chromosomes during metaphase 1. Number of gametes 2 to power n, where n are the number pair of chromosomes. Human 2 to power 23 gametes. Second reason, crossing over between known sister chromatids of homologous chromosome in meiosis 1 prophase 1 pachytene. Random fusion of gametes during fertilization, multiple possible ways. Cost of sexual reproduction: males have to be maintained. Lot of time is wasted in finding mating partners. Progeny may be inferior to parents. Sexual reproduction has more benefits than the cost. Variations improve, evolve, and surviving chance increase. Sex determination: early developmental decision taken by bipotent embryo, followed by sex differentiation, this decision point. Environment: on the basis of environment, according to environment, temperature decide sex in reptiles, surrounding hormones, borrelia. Ploidy level: haploid male, diploid female in honeybee and wasp. Genic balance: number of X chromosome by set of autosomes. अगर 1 होगा तो फीमेल, 0.5 होगा तो मेल, ड्रोसोफिला के केस में. क्रोमोसोमल बेसिस: XX फीमेल, XY मेल ह्यूमन, XX फीमेल, 0 मेल ग्रासोपर, ZZ मेल, ZW फीमेल बर्ड्स, ZZ male, Z0 female ducks and proteinor. Humans sex determination: if 44 plus XX, 44 autosomes, XX allosomes or sex chromosomes. Genital ridge: genital ridge convert to bipotent gonad in the presence of SF1, WT1, LHX9, GATA4. Bipotent gonad to ovary in presence of XX, FOXL2, RSPO1, WNT4। WNT4 प्रेजेंट होगा तो वो इनिबिट करेगा फॉर्मेशन ऑफ टेस्टिस को. Ovary then have granulosa cells and thecal cells. These two cells form follicles. Follicles into estrogen. Follicles secrete estrogen determination. Mullerian Mullerian duct is converted to female internal genitalia, uterus, oviduct, cervix, upper vagina in presence of estrogen, and genital tubercle, urogenital sinus convert to female external genitalia, labia, clitoris, lower vagina in presence of estrogen. Formation of female internal genitalia and external genitalia is differentiation. It is differentiation. If 44 plus XY, then Y is sex determining region of Y chromosome SRY. Genital ridge convert to bipotential gonads in presence of SF1, WT1, LHX9, GATA4. Bipotential gonads convert to testes in presence of XY, SRY, and SOX9. SOX9 inhibit WNT4. WNT4 नहीं होगा तो ओवरी नहीं बनेगी. टेस्टिस के पास दो सेल्स हैं, सर्टोली सेल्स एंड लेडिक सेल्स. सर्टोली सेल्स convert to anti-Mullerian hormone AMH in presence of SF1. Leydig cells secrete testosterone in presence of SF and testosterone into DHT. Mullerian duct convert Mullerian duct regression or degenerate in presence of anti-Mullerian hormone, and Wolffian duct form male internal genitalia, epididymis, vas deferens, seminal vesicle in presence of testosterone. Genital tubercle, urogenital duct tube convert into penis, prostate, scrotum in presence of DHT. Sertoli cells secrete anti-Mullerian hormone. Leydig cells secrete testosterone, and testosterone convert into dihydrotestosterone DHT. Gametogenesis is the production of male and female gametes, haploid cells. Committed cell do meiosis and form sperm and egg. The primordial germ cell PGC is the primary undifferentiated stem cell type that divide by mitosis and will differentiate toward gametes, spermatozoa or oocytes. It is decision made during early development. C. elegans has P4 cells, ventral, behave as PGC, primordial germ cell, and has Z2 and Z3 cells, founder cells, then spermatogonia or oogonia. FM3 FEM3 required for male development. If FEM3 absent, then gonad convert to ovary. Drosophila PGCs, primordial germ cell, migrate to gonads. PGCs in Drosophila are posterior pole cells. Location of primordial germ cells in C. elegans, posterior ventral side. P4 convert into Z2, Z3, Drosophila posterior pole. Primordial germ cell migration in mouse or humans. 8th day, posterior apoblast of blastocyst. After that, migrate to endoderm, then migrate to gut, then to genital ridge, and then to testis ovary. Retinoic acid, vitamin A. It is secreted by mesonephrons, kidney cells, determine the timing of meiosis and sexual differentiation of mammalian germ cells. In female, timing of meiosis in the embryonic condition. Retinoic acid, then STRA8. WNT4 activates STRA8, then STRA8 do meiosis. If nanos 2 is added, it inhibits STRA8 gene in embryonic condition. Nanos inhibit STRA8. If STRA8 is inhibited, then no meiosis. Males में क्या होता है? No meiosis up to puberty, but at puberty, retinoic acid is synthesized in the Sertoli cells and induces STRA8 in stem cell spermatogonia. FGF9 inhibit STRA8 up to puberty, that is spermatogonia. Up to 13.5 days, retinoic acid is degraded by enzyme coded by CYP26B1 gene. आफ्टर दैट नैनोज 2 एंड FGF9 जीन इनहिबिट STRA8 अप टू प्यूबर्टी। STRA8 जीन एक्सप्रेशन होगा तो म्यूसिस स्टार्ट हो जाएगी।